Good morning. Welcome, everyone, and thanks for coming here despite the rain. It is a great pleasure for me to introduce our distinguished speaker, Professor Mu-Ming Poo. I think you all know Mu-Ming. I don't really need to give a long introduction. Professor Poo got his bachelor degree from National Tsinghua University in physics, and got his PhD degree in biophysics from Johns Hopkins University. And he was faculty at several academic institutions in the US, starting with UC Irvine, then Yale, then Columbia, UC San Diego, and then UC Berkeley. He had a very distinguished academic career. He serves as the founding director of the Institute of Neuroscience since 1999. Professor Poo is a distinguished scholar, and he has made seminal discoveries in neuroscience, including signaling mechanisms in nerve growth and guidance, synaptic modification by neurotrophins and electrical activity, etc. And because of his distinguished achievements, he was elected to the U.S. National Academy of Science, Chinese Academy of Sciences, Academia Sinica, and he's also the founding director of the Academy of Sciences of Hong Kong. Member, right. So he has achieved a lot in his career, and he received numerous awards and honors. And this picture actually I took for him when he received the prestigious Gruber Neuroscience Prize at the Society for Neuroscience meeting last November. Again, this is just one of many awards that he has received. He is well connected with HKUST, in case you don't know. He was involved in the planning stage of our university back in 1989, around that time, right? And he was the honorary degree recipient in science in 2014. A doctorate honorary degree from our university. And he's currently the IAS visiting professor. And today, I think you can go back to the title. So he would like to share with us his thoughts on the China Brain Project and the future of Chinese neuroscience. And for the China Brain Project, it started more than 3 years ago, I think. We had our first meeting in Hangzhou, and through the past 3+ years, Professor Poo has been leading this effort in planning and also in coming up with a framework for the China Brain Project. So he would like to share his thoughts with us today. So please join me in welcoming Professor Poo.
Thank you. Thank you, Nancy. It's a great pleasure to be back here. I think this is my 4th lectures at the Institute for Advanced Studies since last year. It's not going to be the last, but this is the biggest sort of grand talk on this big project. Having involved in the organization, I have a lot of thoughts about these projects, and this project is going to be initiated soon. As you know, there's a major drive in the last couple of years to come up with a major science and technology project in China for the next 15 years. And there are 6 science projects and 9 engineering projects approved for 2017 to 2030 by the People's Congress last year. And this is the so-called 6+9. Among the 6, there are 4 is already on the on the final stage of approval and initiation. These are 4 science projects. There's a space-land unified communication systems, quantum computing and communication, and brain science and brain-inspired technology ranked 3rd in this 4th project. The last one is deep sea research. We are hoping that by the end of this year, the formal go-ahead will be announced. So today's my thought will be concentrate on this. I say it's personal perspective, so it doesn't reflect the majority probably opinion, but it's my personal opinion, what I understood, what's going on. So the formal name of the project is Brain Science and Brain-Inspired Intelligence Technology. It's a project with a central core of understanding neural basis of cognitive function, and this is the basic thing in neuroscience. And to do this, there will be a development of brain research technology platform in support of this endeavor. And as 2 sides of these projects, we call the 2 wings. One is the applied wings of the projects, and it's to develop effective approaches in early diagnosis intervention of brain disease. And the other wing is to develop brain-machine intelligence technology. We call the brain-inspired intelligence technology or brain-machine fusion technology between machine and the brain. And this is a scheme called One Body, Two Wings with the central pillar in basic science. What is the understanding of cognitive function? There are 3 types of cognitive function we can think of. In fact, we could categorize. One is our cognition of the outside world, the brain function. This includes our sensory perception, multi-sensory integration of the information, attention to the outside world, selective attention, learning and memory of all the events, categorization, decision-making. All these are dealing with the outside world, the capacity of the brain. Now, many of these processes can be studied in animal model system, ranging from the lowest C. elegans, Drosophila, to mice, to fish, to monkey, to human. Right? This is a cognitive process. Many of them are conserved mechanisms. There's also the cognition of self and non-self. We know we are different from others around us. This self-awareness or the ability to have empathy, to sympathize with other people, to have similar thoughts as other people. A theory of mind of other people. A theory of mind means we understand what other people are thinking. All this requires you to know yourself and others. Right? This is the basis for social interaction. And this is not clear how this can be studied in all the animals. Many people think that this would happen best to be studied in non-human primates because they are closest to human. They are probably more likely to have self-awareness and empathy as we understood it. So there are also another level, the highest level, I would say, the cognition of language. This is human unique. The vocal communication is universal with many animals, but the communication with syntax, with grammar, open-ended construction of sentences, all this ability, even the closest relative chimps could not have it, could not be trained or learn it. So it is unique to humans, but how do we study it? We can study it in cases of human patients with damage of brain that cause language disability, but to really for experimental system, we need an animal system. So far there's no animal system. In the past, people thought maybe the songbird would learn to sing songs by imitation. Songbird may have sort of rudimentary language ability, maybe learning how the sound learn to sing songs may tell you something about the origin of language. So that's been one animal model for studying this. But there's a possibility that the reason a human brain is different, a human have a language, is that we have a different genetic the basis. And the human unique genes define the brain that are capable of doing language. So having human gene identified that are important for language, if this gene are introduced into primates or close relatives, and maybe we can develop a transgenic primates that would have rudimentary ability, or will have the ability to learn the language, right? So there's a possibility now doing transgenic monkeys, which people are now trying to do. One might to introduce human specific language related genes, right? So this is one possibility. So these are all the 3 levels of cognitive function that we need to understand. Now that's what we call understand the basis. Now many of the current studies around the world are mostly dealing with the first level, the cognition of the outside world. They're very advanced, mostly using mice, rodent as animal model system. But the cognition of self, non-self, and cognition of language, these are the higher level cognition that poorly understood, and that will be the future. And in fact, 20 years from now, when we solve all the problems, exhausted all the problems on rodents, then the higher level problems we have to deal with. So this could be the future for neuroscience and maybe future for Chinese neuroscience as well. So how do we have a complete understanding of cognitive function? To have a large project, national project or mega project in many other countries around the world, people thought that organized effort would be helpful to deal with problems that cannot be solved by individual lab, even though we can do experiments to do different studies on the cognitive process. But to complete understandings, we need to understand the entire structure of the brain. We need to map out the structure of the brain before understanding. So there are some mapping problems. We need to map what are these different cell types, because the brain are formed by networks, and these networks are formed by different cell types. So we need to know how many cell types are there in the brain. What are their properties? Right? Or we know, it's hard to believe that we don't even know all the cell types in the mouse, in the rodents. Right? There are probably hundreds. There are probably even more. So efforts being made to characterize the cell type. Once we have the cell types, specific cell types identified, we can specifically label them with the markers, then trace their connection. So knowing all the connection would be useless if we don't know what are these connections made by, or which type of cell are making the connections, right? Inhibitory neuron or excitatory neuron, or what type of subtypes. So connectivity map depends crucially on our cell type map first. These are being done right now in rodents, very rapid pace. We call this connectome, right? The connectome of all the nerve cells or neurons in the brain. Once we get the structure done, that's not enough, because we need to know the function, how they work. Then we need to map the activity. So when specific connections are worked out, then we want to know what type of pathways or circuits is responsible for carrying out various cognitive functions. Okay? So to relate the activity of the neural circuits to the cognitive function, we need to map the activity during the function. And that's a difficult problem. And these problems, this activity map can be done right now in small animals like a fish, zebrafish, where there's transparent brain. We can look at their activity for all the neurons in the brain, so we can carry out mapping for these particular animal models. But it would be it's difficult to do that in mice. For mice carrying out cognitive function, immobilize the head, putting electrodes, putting optical detectors into the brain, map the activity. That's the type of effort. Most frontier work currently going on in the field. So knowing the map, then we can understand how the genetic programs determine the brain building blocks, right? How the cell types comes about. What are the molecules responsible for different cell types? What are the different circuits being made? How the particular proteins and molecules expressed in the cells that generates the circuits, and how that circuits produce the brain functions and the cognitive behavior. Right? So from mapping to understanding is the systematic, complete understanding that would require this. Although without the complete mapping, you can still do piecemeal of the circuits. Do mapping. It's like trying to feel the elephant by touching blindly, right? What the elephant looks like. But complete mapping is eventually required. So to do all this work, there will be brain technology platform required. One needs to do neuronal label with specific cell type in order to trace their connections. We need a brain imaging technology to map out the connections. We also would need functional recordings for monitor the activity, physiological, electrochemical recordings of the activities in the brain. To know the causal relationship between the activity and the function, we need to manipulate activity, see what happened to the function. Right? This is required for causal relationship. To determine the causal relationship, we need to have neuromodulatory methods. We need to be able to manipulate specific proteins, specific activities of defined cell types. So all these are technology that's been on the frontier in the neuroscience. And large data collected needs to be processed. And we need good animal model systems to study higher cognitive function. As I mentioned, we need to have a transgenic or primate platform. This is actually rather unique. I'll come back again and again today on the use of primates in the China Brain Project. Now I should mention that the United States has a brain initiative. Its main focus is to develop technology. It's a mega project by the government to develop new technologies that would help to understand the brain, right? Help to map the brain and understand the brain. Right? So the China Brain Project will also will have to work on this, and in order to pursue the study of the basic neuroscience. For the applied RIN, I would mention briefly today about brain-machine intelligence technology, what it means. First of all, it's the brain-machine interface, or also called the 脑机接口, the BMI. The BMI is critical for medical applications, for neuroprosthetics, for interaction of human with the machine. This would be part of this project. We also need development of different modulation technology. This is very useful for brain science study in brain science and also in brain disease therapy. Electrical, magnetic, ultrasonic neuromodulation technologies. And these technologies are being developed in many parts of the world right now. And this would be one of the focus. And then the most important part of this apply wing is the brain-inspired computing methods. Right? And, as you know, that there's a great surge of interest on the artificial intelligence, the application in various aspects of technology, and the current machine learning algorithms and methods are energy requiring high energy, and it's actually low efficiency, although the capability is strong. Now, AlphaGo, who won the game, or the Go game with the master over the Master Li, is a huge computational success. Right? It requires huge supercomputer to do the rapid learning in order to make the move. So the current deep learning network, for example, are in a way a very inefficient learning network. One needs to learn from the brain. The brain runs at a 20 watt power, which has a capability much better than a supercomputer. Right? You look at this TV show that the Master Li is making move, and the opponent is not a machine; it's a human. The human learns from the instruction from the computer and make the move. Right? Why can't a computer? Why a robot makes the move? Because our robots or all the other aspects, except the computation, are so poor. You have to have a human to help. So, so actually, it's not a machine that went over Master Li. It's the machine plus human. Right? So we need a new generation of computing architecture that can learn from that one needs to learn from the brain. Now there are many aspects of the neural circuits, the natural neural circuits, that are not incorporated in the current machine learning or deep learning network. There are some aspects. Deep learning networks many layers. That is actually inspired by there are many serial layers in the visual system, in the processing of information. But that's all. The connection, the way the connections are made, the way the connections are modified by experience are totally different from what brain is doing. So there's another, on the other occasion, one of my previous talks, I mentioned many of the features of neural circuits that can be applied. So these are the central focus, actually, of this apply ring, is to develop new architecture for artificial neural network computing. And then also the processing of storage device, the hardware for the AI that are brain-inspired, the neuromorphic chips, what people are talking about, that you can simulate what neuron is doing on the chip using the new technology. So these are finally, what strikes the imagination most is brain-inspired robotics. Can we produce a robots that behave like a human, that can serve the function as a human? So this is the ultimate output of all the brain-inspired technology, is to have something that really can serve as a human, with a human capacity to serve a human function. Right? So these are all the interests of all the AI people, or at least a major portion of AI people are interested in this type of work. And so this would be part of the brain project. So unlike the, perhaps closer to the European Human Brain Project, that there's a strong components of translational into technology, right? This is, in the China Brain Project is sort of, I would view it as a more balanced, broad spectrum brain project, have both the basic science as well as apply, and apply in both the neuro disease as well as in technology, right? It's I think it's a balanced scheme. So about the other wing of application, the early diagnosis. In fact, there are debates among the community in China that there are many neuro diseases that needs to be addressed, right? But why is it focused on early diagnosis and early intervention? Now, it is an opinion of many people who are organizing this project, feel that the brain disease disorder is being studied all over by many programs. If the China Brain Project needs to have one specific goal, that goal should be set for early diagnosis, early intervention. Because the brain disease at this time, the critical issue is how to diagnose early. And once you have the disease, have the disorder, the therapy usually doesn't work as well. The recovery are limited. So the best way of preventing brain disorder is to do it early, to diagnose early and slow down the progression of disease or prevent the disease. So the disorder we're talking about are the following. The developmental disorders such as autism and mental retardation, psychiatric disease, depression, addiction, the disease of the young, disease of the middle-aged, disease of the old, neurodegenerative disease, right? Alzheimer's and Parkinson's. All these are at present no cure, no drug that's really effective. So early diagnosis and intervention would be important. Now, first of all, we certainly want to know the disease mechanism in order to target our intervention approach. Genetic epigenetic factors, the pathogenesis mechanism. What are the neural circuit dysfunction associated with it? Now, once this is a long-term problems. We even for the Alzheimer's and depression or the common problem, autism, the pathogenic mechanism is still under active research. We don't have answers. We don't have a clear answer yet. But before that happened, before we really understand the origin of disease, we could develop early diagnostic tools, right? Because we can relate to the symptoms. Symptoms is clear, right? The output is clear. Every disease has its symptom. If we can relate the early sign of dysfunction to the possibility of future disease, then we can have early diagnostic tools. So these early tools, you can use genetic markers, maybe the genetic composition is different, maybe there are molecular differences in the blood, in body fluid, in the cerebrospinal fluid. You can use as a marker for the early marker for disease, or you can do brain imaging to see a structure or functional sign for the future disease, and also cognitive functions. And one can see whether the cognitive function has early signs that are predictable. Now we see the Alzheimer's. Usually you have the Alzheimer's. You detect it, that you have Alzheimer's mostly by very severe functional defects. Now, this functional defect, like memory loss, that's the most clear sign. But this memory loss might have early signs, memory deficits that can be detected much earlier. But there's no study to have that very thorough longitudinal studies to trace out the really earliest sign for the memory decay. Right? You need to longitudinal monitor of this function. Right? So this is a cognitive marker is not well used because there's no people don't go to test memory function early, right? Before they have problems. So once you know once you have this marker identified, then you can say, well, what kind of approach we can do to intervene with the progression of the disease. You can do exercise, you can do memory tests, you can play mahjong to help your memory. There are all more well-designed memory training protocols that you can help you to maintain your memory. Things like that. That's a physiological or physical modulation. You could have a pharmacological memory affecting drugs or other functions as well. Pharmacological preventive drugs. And finally, in order to do the intervention, you got to have animal models that are very close to human. Have neural circuit structure and function very close to humans, so that you can test your intervention tools before you apply to humans, right? So this is absolutely necessary to have non-human primates that has a disease symptom. You have a non-human primate model for the disease, right? So there are two types of monkeys, the cynomolgus and the rhesus monkey, 恒河猴 and 石蟹猴. Rhesus monkey is most often used, but cynomolgus is used by pharmaceutical company a lot as an animal model. So, but very few human brain disease model has been developed. The best known disease model is the Parkinson model, which is a drug treatment that kills your dopamine neurons and produce symptoms that are somewhat close to Parkinson's. Then one can test the drugs on these Parkinson disease monkeys. This is the only widely used monkey models. No other disease have monkey models. So there's a huge area that needs to work to produce them. There are challenges and opportunities. There's a large population, patient population is a big challenge. It's also an opportunity because you can collect data, right? There's a rich resources of Chinese medicine, right? That you can get ingredients out that are available with for brain treatment. Nancy's recent development of a potential AD drug is a good example. Abundant non-human primates in China. China is the biggest monkey producing country in the world. It's mostly for exports to the US and Europe for drug companies. And the export is actually dropping. So there's large resources for monkeys. There's large population of scientists as well. Now there's a huge influx of returnees from abroad in all areas, including neuroscience. And they are well-trained technicians. So the personnel, the human workforce is large. But there's a poor standardization in clinical data and in data collection and in the analysis part. Imaging data or the criterion for particular disease. All these are not standardized, so large patient size doesn't have advantage if they are not uniform. Okay? So in order to have advantage of a large population of patients, one got to have better standardization. Genetic studies are also limited in China. Most of genetic studies are done in the Caucasian populations. They are studied to have a Han population data now coming out for various brain disease. But there's no definitive results on the genetic characterization of various disease Han population. There's a lack of long-term collaboration because for large data sets, one need to share the data from different clinical centers and research institutions. So there's a need for organization at the national level. That's why the China Brain Project can play a role. Now, finally, there's a good advantage that China has a tradition for social mobilization. If government really wants to do it, they can mobilize people by various means. And that's the advantage. Now, if you want to go to the moon, we'll go to the moon. If you want to eliminate a plastic bag in the supermarket, well, in 1 week, nobody uses it, right? So that's a good social mobilization. Clinical platform are required. This is a national level, must be established. National database for brain imaging, unified brain imaging is a big problem. Brain imaging machine, different centers have different machines, different image processing capabilities. So all this needs to be unified. A national blood-based biobank for brain disorder, and also the brain bank. Now, China has the largest population in the world, but the best brain bank, if you want the brain preserved after death, biopsy, autopsy, after autopsy, the collected brain in a standardized way to preserve in a usable manner for the investigators. Best brain bank is from Holland, the smallest country in the world. Right? Even Chinese investigators have to order brain samples from Holland because there's no brain bank in China that's well standardized and useful. So we need that. Right? A national brain health training and education center. So one premise of this brain project is that even though our current understanding of the brain is still superficial and incomplete, there are already many valuable contributions one can make to the treatment of brain disorders and brain biotechnology. So go along with this is the drive for Healthy China 2030, President Xi's big thing announced last year. The brain health 2030 will be important part of it. Right? You got to have a healthy brain besides healthy body. And so the healthy brain development and maintenance, maintaining normal function of aging brain, preventing a delay of neurodegenerative disease, these are all high on the list. It must be done. Hopefully by 2030, we have some progress has been made. Look at the Chinese age population. Currently, 130 million aged before above 65 years old. By 2030, we'll see about 230 million aged people, right? This is a huge population, and they are living very well. The average age is 76 years old. So they are well going to the age that will have Alzheimer's, Parkinson's. So this is a problem that we have to deal with. So let me give you an example that what we can do as basic scientists. So I just mentioned that we need to monitor the brain function early, so we know that bad things are coming. Right? There's no good broad spectrum quantitative tools for measuring brain functions. So over the last years, we in our center in Chinese Academy, we are working to develop broad spectrum, easy to use, video-based quantitative assay of 10 functions, 10 brain functions that can be done within 1 hour, including a simple EEG and eye tracking device. Right? We call this brain health checklist that could possibly introduce soon into the regular health checkup that everybody will go through, especially beyond middle age. Right? We do our health checkup, check everything in our body except our brain. Right? We have a quantitative measurement of our blood pressures, of our blood sugar, but we don't have a quantitative measurement of our memory capacity. So these 10 functions, So 13 laboratories, all basic science laboratories. Everyone contribute a piece that they are best, most good at. Expertise together to bring these 10 functional tests. Right? This includes the sensory functions, all the brain function, sensory motor functions, attention, decision-making, and memory, different type of memory, working memory, or declarative memory, and also social aspect, language processing. This would be accomplished within an hour and a half. And right now, the last version hasn't been applied yet. So if this is introduced into a large population over a 10 years period, then we can monitor quantitatively the change in various parameters to determine early diagnostic parameters. If you go into the clinics for your neurological or psychiatric problems, what you get is a simple questionnaire, a list of 10 questions, how good is memory, do you forget anything, things like that. The doctor will check the list and decide whether you have Alzheimer's, whether you have bipolar, you have schizophrenia. All this qualitative diagnostic tool is what recommended in the latest handbooks called DMS-5, Diagnostic and Statistical Manual. Every physician go through the DMS-5 to check out the list. And all these symptom-based diagnostic tools are unreliable, subjective, they overlap among all the diseases. So there's a great need in the medical community to have an objective criteria of diagnosis of brain disorder. In fact, in the end, I think 20, 30 years from now, we should get rid of the label of depression, compulsive disorder, or bipolars, or we should eliminate all this label. We should mark the problems, the brain problems, by their defective functions, right? You have a sensory deprivation, sensory perception problem. You have a working memory problem. Your functional problems, it should be the target for your treatment. So in the future, a clinic for the brain disorders, you go in there, you do the test, people would give you a treatment, a drug or a intervention, physiological intervention, prescription that are aiming at a particular function, just like lower your blood pressure. This will increase your working memory. This is what we need in the long run in the therapy of brain disorder. So let's come to back to the Brain Project. The China Brain Projects offer this opportunity so we can have a big progress on problems that can only be solved through large-scale teamwork. Now, the example for this is the Human Genome Project. Right? The Human Genome Project is what people think that's very useful, having the complete sequence of the DNA of a human. And then people get together and did it. No single lab can do it. This required a large project. The same is true for the connectome. If we want to understand the brain, ultimately it will be very useful to have all the connections among all the different types of cells in the brain. That connectome is required. If we have that, just like we have a human genome sequence, we can do a lot of analysis. We know where to look for problems. Right? It's a database that the field needs. Right? Basic science needs it. Clinical people needs it. So Brain Connectome Project is very important. Now, in the US and Europe, people are working on this. There's a Brain Connectome Project on a macroscopic level. mapping by brain imaging technology that maps the major the nerve fiber tracks, but with a resolution that are not very useful to understand the mechanism. So what we need to do is to map the connectome we're talking about is a mesoscopic connectome. Right? Macroscopic connectome, like what you get from brain imaging data, are this type of fibers, right? Even though these fibers are deduced from the magnetic resonance imaging data. Each of these lines constitute thousands of fibers of different type, right? You don't really know what it means. In fact, all these tracks, some of are based on the mathematical models in treating the data, right? So it's not necessarily reproducible and reliable. So what we really need is the mesoscopic level, at the micron level, at single cell level. So if we have this labeled cells labeled a few cells in the piece of brain, right? When only a few cells are labeled, you can see their fibers already so complicated. So if there are fewer cells, or if there are all the neurons are labeled, this is a mess. You couldn't see it. Right? So in order to see this, their process, we have colored each neurons with artificially with color. But we don't know their cell type yet. And for example, in the brain, in the cortex, the same type of morphology looks similar cells. They may be different type. They have different input-output connections. So you can go even deeper, microscopic level, submicron level, where you can know all the details of connectivity at the local regions. That's also the complete information if you really want the connectivity. But this is too complicated. Right? To get a small piece of brain to get this microscopic connectome, it takes too much effort, too much reconstruction work. And so the most realistic and useful level is the mesoscopic connectome. Right? Knowing the cell type, if we know all the connections, the input and output connections of every cell types in different brain regions, then we have that's the connectome is most useful. So to do that, we need to do this. So there's a general consensus that we will, within the China Brain Project, we will initiate a Connectome project. Final go-ahead for the monkey. The final go-ahead for this is not yet arrived. But if we want to have a large-scale project that would be international, that China would play a major part in the international collaboration, like a particle physics experiment, where people are grouped together internationally to work on an important problem for the field. We think macaque brain or the monkey brain connectome is this type of project. It's like human brain genome project that could bring people around the world together to solve it. It is not a single country or a few laboratories can do this. It needs a large group of effort, right? So hundreds of people, hundreds and thousands of scientists might need to do this because it involves first figure out the gene expression and the cell type map for all the region of the macaque brain. Now you want to figure out with molecular technology, molecular biology technology, in situ hybridization, and single cell sequencing. There may be thousands of different types in the macaque brain. That needs to be figured out. Once we have the specific type, then we have to label each type and find their mesoscopic connectome, map their input and output of each type of neuron in all the brain region. Then once that's done, maybe 20 years from now, we can further look at the activity map. Technology is mature enough so you can put a probe into the macaque brain. You can ask the monkey to perform a function, and you look at hundreds or thousands of neurons, how they fire during that function. Then you to figure out the logic of neural circuit, how they perform the function, right? So this is a project for decades, much longer than Human Genome Project, hundreds times more difficult than Human Genome Project. But this is something we will need, or we really in order to really understand the brain. Right? So why focus on non-human primates? Because to understand the higher function of cognitive function, as I said, there are 3 levels. The important higher cognitive function of self and non-self and language, all these are only appear in non-human primates and in primate systems. So to know the neural basis of the higher function and also how they evolve during evolution. Now, most people justify their study on model animal in mice or in C. elegans in worms because there are mechanisms that are conserved. Whatever you learn from a lower animal systems can be used to understand higher animal. That's the rationale of conservation. That mechanisms that are conserved, indeed, there are mechanisms conserved. Firing of action potential in neuron, the neuronal firing, the coding of neurons is the same way in squid, mice, and in human. But the circuit are very different in different species. So there are conserved mechanisms. There are also diversification during evolution. Right? In humans, the essence of biology is in fact diversification. During evolution, new species appear with a different function. And that different function in the brain function reflects different circuits, different type of neurons that have been evolved. So we need to understand the brain, we need to understand both the conserved mechanism, also the diversification mechanisms. So that's why ultimately we need to have a primate, from mouse to primate to human. We need to, this will facilitate to the understanding, the pathogenesis and development of diagnostic and therapeutic tools, right? Because primate is closer to human. It's the best animal model. Also, this is very important. I think the most important part of using primate is to help to dissect the true meaning of macroscopic imaging data. Now, you go to the medical clinic, you can do brain imaging. If you, the most useful brain imaging diagnostic is for where the tumor is. It's big structural deficit, MRI, PET scan, for big changes in your structure of your brain. That's why brain imaging is useful. You want to go to imaging, take an imaging, you want to figure out whether you have depression, whether you have Alzheimer's. That's much more difficult. Now, for Alzheimer's and Parkinson's, now there are PET imaging that can be helpful. But for most of the other disease, psychiatric diseases, no, brain imaging is not useful because we don't understand fully what the macroscopic brain imaging really means in terms of neural circuits, right? So having, in order to understand that, you need a model system where you can manipulate the circuits and look at the corresponding changes in the macroscopic imaging. So in a cat brain, you can look at the mesoscopic level, the circuit level. You can have a macroscopic level. So you change the circuit level phenomenon, you look at the macroscopic level. So you can understand when you have a signal like structural imaging, DTI signal, and they go wrong, different. You understand what it means. Right? So this is the most important part, I think, to have a primate to study this. I think my time is rather limited now. I probably should stop in 5-10 minutes. Okay, yeah, around that. So, I'll move fast. I think a big brain project mapping all the type in the cell. This is an example of the cell type. 100 years ago, Cajal, a Spanish anatomist, already start to do this based on morphology to distinguish cell type. But the modern technology says, well, even the cell with the same shape or same morphology, they express different protein. They have a different genetic endowment. They have different function. So you need to have a functional as a genetic criterion for cell types. For example, you can for cortical neurons, take a piece of cortex. You can do the gene profiling. It's called the RNA-seq, deep sequencing. You can look at a profile of gene expression. Then you can cluster analysis, put them in a group, the similar patterns grouped together. You can make, for the cortical neuron, you can easily distinguish 47 different cell types, even in the cortex. Now, most of us knows there are about 5-10 types before, but now gene profiling tells us it's much more. Now, this is still not the end because this is how they cluster. They define the cluster by similarity in the profiling, right? So if you have a better profiling, you can subcluster. You can make more clusters out of this. Then you can define more types, right? So by genetic profiling, there are there are hundreds, maybe even more, that will really appear. But how do you end? How do you say, well, this is the real profile. This profile defines the cell type. That's a difficult problem. So now there's I have proposed a definition of cell types, which have been discussed about among our colleagues who are interested in this problem. Is that the cell type should be defined not only by genetic program, which is early development. You have major neuronal type determined. It also type are determined by connectivity because there are interaction between cells after the circuit is made. So subtypes are developed because of the defined connections made through the cell-cell interaction. So in order to determine the type, the subtypes, one needs to determine the input and output pattern of each cell, even with the same profile, similar profiles. Right? So it is the subtype should be distinguished. Actually, it's a mistake. The neuronal state and subtype should be different. The state is the same type with a different expression pattern, but that's dependent on the state of a neuron. They can change with time, right? So one has to distinguish the subtype, which is stable, and the state of the neuron in their gene expression pattern. So our definition is that neurotype is best described by a combination of expression pattern and the input-output pattern. That's why the single-cell connectome is required. So, for example, this is a tracing of cortical neurons. They're all similar shape, but they project to different areas, right? So if we consistently see that some neurons project to a particular areas, they should be considered a different type. Because if you look at a gene profiling, you could find specific proteins that are more expressed in this cell. This is just colored without really single cell identification. This is a Thyroid neuron, Thyroid mark. So this is the type of work that needs to be done. So finally, a slide. There are many problems. I would maybe leave this for the next lecture. I have half a lecture that going about what I think is the major problem in neuroscience. I think the major problem, now everybody has their own list, but most of the lists are all overlap with each other. So my list probably overlap with most other neuroscientists. So I say the mechanism of underlying learning memory. We don't know that. The binding problem, how do you bind different informations together? Now, different sensory information or different type of sensory or different features within the same sensory information. Right? So this is called the binding problem. How do you, what is the sleep for? What is circuit controlling? What is dream for? And the neural basis for social affiliation. How do you social with people? How do you establish self and non-self and your identity? How the empathy, why do you have a sympathy with others? Neural basis of consciousness. And this actually runs top on the list by many people in the list. And I think this is not so mysterious. People think consciousness is very mysterious. I think consciousness is just as mysterious as of sleep, because it's a different brain state. So it's not I'll talk about this in the future. This is an interesting topic. Evolution and the neural basis of language. So that's the major problems. 6 unsolved problems. I said 6 because I read a book. Chuck Stevens wrote a book on the 6 major problems in physics or something. Okay. Finally, I should go to the end. This is a thing that I skipped for the next time. I would say that there are issues coming with the China Brain Project that people could be aware of. Now, this is a long-term project, so the government is thinking, and most people feel, some people feel, at least myself, feel that the reform of the structure of a science and technology management system and mechanism should go along with the big project. This is the opportunity to reform, because without reform, the project would not work well. Right? So the key is the institutional evaluation system, which I can go into deep more further. Without changing the institutional evaluation of the investigator and in the evaluation of the accomplishment of the project or a scientist, you cannot get the big teamwork to work. People will not collaborate wholeheartedly to get things done. Right? This is the key, and the evaluation system is in deep trouble in my mind around the world. Now, scientists now, young scientists, are reluctant to collaborate because they are evaluated based on independent contributions for their tenure, for their grant, for their prizes, for everything. Right? So that prevents a big. Now, the problem of evaluation has been solved in physics. Right? Because they have many years worked out a system that you can be part of a project with hundreds of people, you get recognized properly, but not in biology. Right? So this is a problem that we need to deal with. Small science versus big science. Is there a conflict? If you want to do small science, is there a conflict with big science? Like the mega brain projects? I don't think so. I think there's a way that both can go together. Right? So, I don't have time to go into that. Independent network are going. You need even the big science need innovation. Right? That's happening in physics. It will happen in biology as well. Pursuit of individual interests and societal needs doesn't necessarily to be contradictory. Right? There are fundamental issues that one needs to consider. Is the role of scientists in the society. What are they for? And where is the science, Chinese science going? How can Chinese science be unique? Can Chinese science make unique contribution rather than just catching up? I think so, but I don't have time to talk about it. So I'll stop here.
Thanks, Professor Poo, for your sharing of your insights about China Brain Project and neuroscience. So generally, I have 2 small questions. The first one is that for some neuropsychiatric disease like anxiety or depression, they are pretty circuit specific, and there are circuits in the brain counteract with each other. Some are promoting the anxiety, some are anti-anxiety. And for the currently available drugs, most of these drugs are the antagonists or agonists for some receptors. And more research using the optogenetics has found that it's like very circuit specific. And people like Carl Desros has been trying to use optogenetics to stimulate specific circuits. But I think the problem with the optogenetic stimulation is that it's too invasive. So what do you think?
Right. So that's why we need new technology, invasive or non-invasive. If we can target specific circuit, if we know the specific circuit underlying depression, or anxiety. If we know specific circuit, that's people have ideas that what happened in mice that a certain circuit might be involved, but that's not only circuits. It's not unique yet, right? So if we really need a important circuits that are important for a particular function, one need to develop specific modulation tools. Now, deep brain stimulation is being used for severe Parkinson patients, but it's invasive, right? You have to put an electrode deep in the brain. Those are very specific, circuit specific. You target exactly the system that control the movement, right? So can one develop that? We'll develop tools that are more specific in the future. It's possible, and people are still working on it. There are non-invasive transmagnetic stimulation, which are not very specific. There may be ultrasound technology, magnetic stimulation, electrical stimulation, transcranial direct current stimulation. Those are all not very specific. So how do you target specifically is a big problem. That's why there's a technology required. Technology development is.
I have a question about the brain-inspired computational method. Now, this is a big field that has a lot of promising future. Right. But I wonder, how can people overcome the challenge of connectivity in the hardware side? Because in the brain, each neuron will make a lot of connections, maybe 10,000 connections with other neurons. But in the man-made circuit, you're limited. You can only make limited, far less number of connections with the other electronic components. So hardware-wise, it's impossible to mimic how the brain functions. So how can they overcome that problem?
No, I don't think this size is a problem. One can build with miniature technology. You can build more connections than the brain. But the problem is not the number. The problem is the efficiency. That the connectivities, the hardware now used are not as flexible, not as efficient in handling information as our own connections in our own brain. Right? So for example, in hardware, you need to the current hardwares you need to store your information in a storage area. Right? Processing area and the storage areas are separate. These are separate, so you need additional connectivity between these two separate areas. In the brain, there's no storage area. The memory is stored in the circuit that processes the information. It's a distributed memory, distributing storage of memory in the circuit, and that's very efficient. So, and also the circuits is not as numerous as you think. The possibility is that each neuron can make 3,000 or thousands of synapses with another neuron, right? It can receive thousands of synapses, but not all the synapses are being used, right? And the synapses are actually modified throughout the experience, constantly being modified, right? So I think one way to look at the brain circuit, people think that we can make decisions, human brain can make decisions based on small data sets, while a computer needs big data sets to learn. But that's not exactly right, because our brain, our circuit, actually has used a large data set through our life, modify our circuit, right? From the day of our birth, our circuit has been constantly learning and to sample all the information in the environment to modify its architecture. So our current architecture is actually learned through much more information than the supercomputer learned from the big data sets. Right? So that's the developmental aspect of it, that the connection actually can be modified, is an important feature of our natural circuits. While the current AI circuits are not modifiable. Their weight can be changed, but the architecture cannot be modified.
It's very interesting that you mentioned connections of a cell
defines his status.
Yes.
That means cell connections.
The connections is very important.
Yes.
So was it common that also this connections of cells is important for other organs like liver or lung? And what's the difference of these connections in liver from the connections in human
In
brain?
connections between liver cells. Yeah.
Yes.
So brain tissue is very special compared to liver and other tissues, right? So more the other tissues first of all they do not make extensive connectivity. Right? A single liver cell makes contact with nearby liver cells, a limited number. They do not have a long range communication with other liver cells within the liver. Right? Maybe they are diffusible substance, but there's no rapid communications or the information are not handled specifically between two cells at distant regions. Right? That's quite different in the brain. Right? That's the first difference. The second difference is that the contact between other tissues, most other tissues maintain their machinery, their cellular mechanisms try to maintain a stable state, the homeostasis. When environmental change, when they receive stimulus, they want to maintain the function in the original state, the homeostatic mechanism. Homeostatic mechanism is key for most tissues to maintain its functional state. But the brain has evolved to a very different mechanism. In addition, they want to maintain their homeostasis, their functional status. They have additional mechanism that's plasticity. They can change. In other words, the environmental input will not only will cause a change in the state of the communication between cells. So the contacts between neurons are modifiable by experience. That's not happening in all the other tissue. Plasticity beyond homeostasis.
Thank you for your nice talk. I have two small questions. As you know, the disease of schizophrenia, the mechanism of its pathogenesis remain unknown, and the animal models for study this disease also have no. So I want to know for in this China Brain Project, does there is any specific goal or long-term endeavor for fight for this disease?
Beyond the early diagnosis and early intervention, for understanding mechanism, it's going to be a long way. Right? We will not be able to understand it within the next 10, 20 years, I would say, the pathogenic origin of schizophrenia. But we can diagnose early. Right? For example, the hearing, hearing sounds, hearing voices, this type of symptoms could be intervened. In fact, one of our collaboration with the mental hospital in Shanghai is to intervene with intervention of the schizophrenia patient when they start to hear voices, produce interfering music to interfere with that sound. Right? Try to see whether one can perhaps physiological intervention could prevent or help to cure the disease. That's type of is the way we can do without understand the disease. And development of an animal model is a very difficult problem. There's nobody have even for mice have a good schizophrenia mice model.
Thank you. The second question is because I have a close friend who is very talented, but unfortunately he has been diagnosed with this disease. And when I stay with him, he is often laugh, and laughed heartily. And I asked him, what made you laugh for? And he said, oh, am I laugh? He has no self-awareness.
Right.
In Chinese martial art fiction, there is acupuncture point, 穴位,
Acupuncture.
call it 笑穴, and the laugh acupuncture. So based on this, I wonder whether the mechanism of this disease pathogenesis is it will unrelated to the brain, or it may be other location of the body.
Well, it has to be controlled by the brain.
Yeah.
Behavior is all controlled by the brain. So we don't know why. Right? So that the mechanism is unclear. So the acupuncture may have, well, certainly will produce effects in the brain circuit, but the way it produces the effect, whether they are effective therapy, there's no, it's hard to say, right? So that's only depends on experience and practice.
Thank you.
Thank you, Professor Poo, for such a wonderful talk. My question is more with the recent reports in Alzheimer's. A lot of pharmaceutical companies have declared failures of very promising molecules. And there are many reasons for that. But before such ambitious projects like the China Brain Project come to fruition, what approaches do you think these drug discovery efforts should basically change? What approaches will they benefit to help maybe the current and the upcoming generation of Alzheimer's patients?
I think the drug development effort should continue, and actually there's a sign that that early application of pharmaceutical approach would be effective. Now, as example is the current success, the sign of success for the antibody treatment. Right? So treating the disease early is always a good approach, right? Because neurodegenerative disease is degeneration of the nervous system. Once it degenerated, to regenerate is difficult. Right? So you want to prevent the progression of degeneration. Right? So early, aiming at the drug development of early treatment is going to be a more fruitful approach. And this would go along with the plan. There's already a plan for large normal and patient population, longitudinal monitor for long-term monitoring. Right? So the drug development and the data from these longitudinal studies are going to be complementary and useful to each other.
I'm a physicist, so let me end this session with one quick sort of physics question.
Right.
If you count how many neurons in the brain, and then you also count how many proteins per neuron, synapse, quickly a network, I think you get 10 to 12, 10 to 15 molecules. And then you were talking about the specificity of the a brain tissues. So I just want to know, a neuron dysfunction or neural network, understanding the function, are we dealing with a strongly correlating many body problems? That means we have to worry about the collective behavior of all the molecules in the network. Or we are talking about in physics called a single body problem. You work on a particular local issues, all the things, because that will affect the ways our thinking. And I just do not know. You indicate different aspects of this thing. So can you sort of comment on the thing?
Yeah, you have to look at it from population correlated activity of hundreds of thousands of neurons. Collective behavior. Emerging collective behavior is just like statistical mechanics. You have to go statistical mechanics approach rather than many body problem approach. Not from the individual constituent. You got to look at higher level emerging properties out of a large number of components.
But that seems to me that the genetics will play a less important role in a way, right? Because that's genetics only mean a certain thing, and then the other part is the connection which matters.
The distance between genetics and the collective behavior on the higher levels are far.
Yes.
The direct connection are harder to make. It's just that, but the genetic does make a difference. Individual molecular properties of the atom or individual atomic properties is important for their collective behavior at the macroscopic level, right, for statistical mechanical properties. There's a relationship, but that relationship is very distant. But it does make a difference. If you have a different molecule, you have a different higher level phenomenon. So different genetics produce different cells. The cell fire differently, right? So if they fire differently, their collective behavior when there are millions of cells fire together would be different. So in that sense, there's a connection, but it's not a direct connection. There's a jump many levels between cells and circuits and many circuits and systems. Right?
Okay, that means that can we sort of do some kind of cross-screen, ignore a lot of unnecessary molecular details and jump on a sort of mesoscale level? You just illustrate.
I say many people does ignore genetics when they look at a higher level phenomena. But if the genetics is wrong, if you want to understand what's wrong with the phenomena up there, sometimes the origin is the genetics. It's like you have one gene go astray. You have autism. Autism is a collective behavior of social interaction defects, but that's due to a single molecule, single gene defects, right? But the connection between that gene defects to explain the autism is you cannot explain it because you have to understand the circuits that gives the autistic behavior. That's the immediate level. So if you want to understand the scientific approach or reductionist approach is that if we have a collective behavior up there, well, you reduce that one level down, you reduce the circuit, understand what is the circuit basis for that collective behavior. That's scientific understanding, right? But if you say about it's very difficult to jump from the collective behavior to gene because there's many missing levels. So the fruitful approach is not to jump across that far, but go step by step.
So, Mu-Ming, you mentioned earlier that the Brain Connectome Project can be an example of a project that involves, international collaboration.
Yes, right.
So, what is the current thinking for the China Brain Project? How it link up with the other projects like the Brain Initiative or the Human Brain Project?
So this the idea of a big international project will consist of two components. One is the international team, like what we do for Human Genome Project. There are many teams taking different parts of the brain to work it out, to map out the cell type of brain. I just like one lab take one chromosome, China take 1% of the genome project. Right? So everybody take a piece. Then that's one approach. The other approach is we have an alliance, for alliance of this particular brain project with alliance of the U.S. initiatives with human brain projects in data sharing. Although they do not directly work on this primate brain, but their technology can be applied. Resources, technology, and data sharing alliance. That's the approach. They were following the Geneva approach that people can actually, if you have a center, if we have a center for mapping brain, a human macaque brain in China, it might attract a lot of people to do the work here. Short-term visits, long-term visits, to utilize the facility. Because if we have models, disease models, well, it cannot be exported to other countries. And the environment for primary research in other countries are not as good. So they would come to this center for doing the work. So that's the way we built a big project.
Okay, if there are no more questions, please join me to thank Professor Poo again.
Thank you.